Good morning. This is David Francis from Ohio State University, and I'm going to give you a brief overview of what the Plant Breeding Working Group has been doing on the Growing New Roots project, which is an SCRI-funded multi-institutional research project. Before I get started, I want to recognize a large number of people from different universities that have contributed to the Breeding and Genetics Working Group. I've listed some of those names here. We have Shakir and Pat from the U.S. Vegetable Laboratory, a USDA ARS group in Charleston, South Carolina. Out at Washington State University, Carol Miles and her students have been very active in screening and developing germplasm. At Florida, both USDA and University of Florida scientists have been involved in screening tomato rootstocks for resistance. My group has been very involved with looking at breeding from a longer-term perspective, trying to identify key selection points. Frank Lowe's and Dilip Panthy at North Carolina State University have been very active in screening, as have Jean Miao and Brenna out at University of California, Davis. So the objectives of the breeding working group were to utilize genetic diversity in Solanaceae and Cucurbitaceae to improve scion health and productivity. I've listed the objective here. So what I'm going to really focus on is the themes, and those are delivering traits through grafting, the use of crop wild relatives or wild species as sources of resistance to soil-borne diseases. I'll talk very briefly about some of the germplasm evaluation that's been going on. And I'm going to highlight the release of a cucurbit rootstock that came out of the USDA ARS group in South Carolina in conjunction with researchers at Clemson University. I'm going to talk some about general principles for rootstock breeding, and I'll just touch very briefly on the emerging issue as we see it of graft compatibility. Okay, so in screening for resistance to soil-borne root diseases, there has been an emphasis on screening for Phytophthora root rot in the cucurbits, so Phytophthora capsicea, for tomato bacterial wilt resistance, so resistance to Ralstonia solanacearum. In both crops, there's been an effort to identify and develop resistance to verticillium, and there's been significant effort and success on the cucurbit side with resistance to fusarium. This everything and the kitchen sink quote comes from Jean Miao, a cooperator at UC Davis. There are numerous soil-borne diseases that may be addressed through breeding, and members of this cooperative effort have screened beyond the diseases that I've listed here, but I think it's fair to say that these have been the main efforts. And again, just to emphasize this, I want to show two pictures, one coming from the group in South Carolina showing a Phytophthora root rot screen in cucurbits, and you can see the devastation that disease can lead to, but you can also see some green plants here and there suggesting that there are in fact sources of resistance. On this picture over here shows some work that came out of Matthews Parrott's lab at University of Florida showing production on rootstock and in the absence of that rootstock here. And so again, the take-home message is that we can identify rootstocks with resistance to soil-borne diseases, and in many cases, those can lead to successful production, where in the absence of grafting to a resistant rootstock, production would not be successful. Just as the sort of sample results, I thought I would put this slide in that was passed on to me by Carol Miles. It emphasizes work that her group has done to screen germplasm for resistance to Verticillium dallii. the measure that they've used, which is area under the disease progression curve, they're using sugar baby as their scion. They screened a number of accessions from the national plant germplasm system and found 18 that had AUDPC values that were similar or lower than the resistant control. Only 6 of those produced mature fruit when grafted to Secretariat. There was no production of Secretariat due to verticillium wilt. And then of these accessions with resistance and no noted failure of grafts, fruit quality differences weren't observed. And then, so the conclusion is the national plant germplasm system contains accessions that have the potential to be used as rootstocks or for rootstock development with resistance to verticillium. And her group has published papers out of this work. So this is just a sample of work coming out of one of the collaborators. So I have a couple of slides that Pat Weckner passed on to me to share with you. So one of the successes of the group working on breeding for cucurbit rootstocks has been the development and release of Carolina strongback. So this slide is showing Carolina strongback grafted with sugar baby and then grown, so grafted and ungrafted and grown on soil that is infected either with Fusarium race 1 or Fusarium race 2. And you can see that the grafted material is surviving very well, whereas the ungrafted material is not. Continuing to move my image here to make sure that you can see everything. Okay. So just to elaborate a little bit on Carolina strongback, its species is Citrulus amorus. It has been used successfully with numerous seedless and seeded watermelon cultivars. It's a proven rootstock for European style greenhouse cucumbers. It has not been a good rootstock for cantaloupe. So it's the result of a cross between 2 selections made by the USDA. Both were selected for high levels of tolerance to Fusarium, both races 1 and 2. It also possesses resistance to numerous other diseases, and I think that's also important to keep in mind. The USDA ARS group has also studied the genetics of this resistance and have identified quantitative trait loci linked to genetic markers. And so that work gives the group a set of tools to further develop resistant rootstocks. Okay, I'm going to shift now and talk a little bit about bacterial wilt resistance. And so I'm sharing some of the results that came out of Dilipanthi's group at North Carolina. Again, you can see the devastation that bacterial wilt due to Ralstonia solanacearum can cause. What this slide is showing you is that across locations, rootstocks have been identified that possess resistance to bacterial wilt. And these rootstocks derive from wild material, but also some of the Hawaii material that many of you may be familiar with. Okay. So sometimes the resistance screening isn't intended, but you end up with a location that has a population of a soil-borne disease, and you inadvertently are able to collect some data. This is a slide of an organic production in North Carolina with a series of tomato rootstocks. The trial had southern blight caused by Sclerotium rolfsi. Again, similar to what was seen with bacterial wilt screenings, there was resistant material. So again, the Hawaii material, including Hawaii 7998, possessed resistance. as did some wild species and a number of hybrids formed by crossing either resistant Hawaii material or wild species. And so you can see that, as an example, Solanum pinelii appeared to have no disease. The Hawaii material is quite resistant. And then a number of hybrids also had resistance. And you can see where the non-grafted and self-grafted controls are here. Okay, so I just want to underscore that is that their resistant material can be identified, that resistance can be passed on through grafting, and often the resistance is coming out of wild species housed in germplasm collections, either the National Plant Germplasm Collection or, in the case of tomato, the Tomato Genetics Resource Center at UC Davis. Okay, so what I'd like to do now is generalize a little bit and talk about some general principles. So again, one of the overall objectives was to address underlying principles that will improve the organization, efficiency, and strategy of rootstock breeding programs. And so as an example, we've already noted that both through cucurbits and tomato, that wild relatives or wild species provide a source of resistance. However, there are issues of working with those wild relatives that I'm going to share with you in just a little bit. And then finally, I want to point out that in organizing breeding programs, we're often concerned about gain under selection and heritability. And I'll explain those as we go forward just a little bit. So many commercial tomato rootstocks are hybrid wide crosses. So that means if we'll focus on this lower picture with the tomatoes, we're taking wild relatives, and I'm showing a green-fruited Solanum habrocates, red-fruited Solanum pimpinellifolium, and then a cultivated tomato. So our hybrid may be formed by crossing this cultivated type to one of the wild species. And those wide crosses often produce vigorous plants. And that's one of the things that I'm showing in the images of the root systems here, is that if we take a cultivated type with this root system, we cross it to a cultivated type with this root system, the hybrid gives us what we would describe as hybrid vigor, a more robust root system. And so that's what we're trying to do with respect to these wide crosses. It's not always as easy as that description may make it sound. There are occasionally issues with uniform germination of seed coming from these wide crosses, and there are occasionally issues with compatibility, which for the purposes of this talk, I'm simply defining as a high percentage of graft failure. Okay, so the other thing that we have to keep in mind is that when we put hybrids together, we're making choices about what to use as a male and a female. Sometimes when we're working with these wide species, we cannot, when we're working with the wild species, we cannot make the crosses in both directions. There's something called unilateral incompatibility, which is in tomato especially common when using the green-fruited wild species. Those really have to be used as male parents. So we're not able to set up reciprocal crosses. We have to make decisions about what we're going to use as males and females. So I'm showing you a partial list of germplasm that we've accessed. Often the cultivated parents were chosen because they have specific resistance or tolerance that we're interested in. The wild species were chosen to represent close and more distant wild relatives, as well as specific resistances known to occur in that material. Okay. So one of the first questions that we set out to ask is whether the genetic distance between parents was a predictor of hybrid vigor with respect to both rootstock growth, but also rootstock performance. So the idea here was if we could use some a priori information, that is genetic distance, it might help us decide which crosses we were going to make. And so this is just showing a partial matrix of genetic distance between 11 parents. The genetic distance is estimated using DNA-based polymorphism spread throughout the genome. there's essentially no genetic distance between parents on the diagonal. And I'm just showing you the genetic distance and the error associated with that in the matrix. Okay. So this material was then grafted, and those grafted plants were evaluated for a number of traits, vine characteristics, yield, and quality. And I'm just showing you a couple of the traits here. These two graphs are showing the genetic distance measurement, which in this case is Ney's genetic distance on the x-axis, and then a trait that was evaluated on the y-axis. And so there's a potentially positive correlation here, so statistically significant, with scion growth in the field relative to genetic distance. So the genetic distance may be the genetic distance between the parents used for the rootstock may be imparting some sort of vigor to the scion. That's what's indicated by this graph. On the other hand, if we look here, if we look at germination rate of that hybrid seed, we see that there's a significant negative correlation. I probably should have switched the position of these two graphs. The point is that there are some positive correlations as well as some negative correlations. And so even though the wide crosses with greater genetic distance tend to give us poorer germination, we can identify individual crosses that give us good germination. And so that's really what we need to figure out how to do, is select for these wide crosses that give us good seed germination. So again, the next point then is to identify selection points in a rootstock breeding program. Okay, and so we evaluated crosses for both seed set and seed quality, for germination, for success in grafting, and then field evaluation. So the idea is, where are the best selection points for rootstock improvement through breeding? And I'm just going to give you one brief example here. So this is looking at germination versus seed weight. And I've superimposed on this graph confidence intervals for 99 and 95% confidence, but I've also superimposed International Seed Federation as well as ASCO cutoffs for what's considered to be acceptable germination for commercial control. And so what you can see is that there is a correlation. It's not a linear correlation through the whole range. So we can do a preliminary screen on seed weight, and anything of very low seed weight, we're just going to throw out without even taking to the germination stage. And then we're going to evaluate material for germination, and we can make selections of wide crosses that give us both large seed and high germination rates, okay? And so rather than taking all hybrids to field evaluation, we can do some pre-screening to eliminate material. And so in this slide, we're just showing you differences in seed weight, keepers, throwers. We're also showing over here that some crosses give us lots of seed and some give us few seed. So even before we get to looking at seed quality, we're probably going to throw some material out just because we don't get a lot of seed. And so in this graph, what we're looking at is a series of hybrids. We're looking at fruit set, we're looking at seed yield, seed weight, and seed germination. And the point is that if we apply cutoffs here, out of 70 completely new hybrids, there's only 11 that we really think we would want to keep to take on to the next point of grafting and then field evaluation. So we can identify selection points that can allow us to rapidly screen a lot of material, and then we can carry forward with a more limited number to graft and evaluate in the field. Okay. So I'm going to try something here. I'm going to try a little pause and take you through a video. I'm going to leave the sound off on this video, but I'll narrate.
Hey, this is David. Let's go for a walk.
field here in northwestern Ohio where a trial is planted. We set up these trials so that we can account for variation that's due to genetics and also variation that's due to the field. When we're trying to select better varieties or better rootstocks, we need to figure out whether we're measuring something that's genetic, something due to variation. We do that by collecting data as objective as we can. And we partition variance. And I'm going to share with you some of the results of that. Okay, so there was a lot of wind in that video, so I'm just going to hit the main points. Whenever we put a field trial in, we use experimental design that helps us to tease apart causes of variation, whether those causes are genetic, meaning a rootstock or a scion, or whether those causes are environmental, position in the field of one location versus another, one year versus another. And so we lay out our fields with an experimental design, we collect objective data, and then we analyze that data. Okay, and so often when we're analyzing data from field studies, we're focusing on a question, is one variety or is one rootstock better than another? So this is what we might call a fixed effect model. And I'm sharing here partial data from a multi-year, multi-location study. So this is a study where we had rootstocks grafted to the hybrid Celebrity. We had them grown in Minnesota, in North Carolina, and in Ohio. And I'm just showing you the rank of where that particular rootstock grafted to Celebrity was in the different environments shown here. I'm using a ranking approach because often there weren't significant differences between, say, ungrafted Celebrity and grafted to, say, this 303 rootstock. I also want to point out that this 316 was the best in North Carolina, but it ranked 7th in Minnesota and 5th in Ohio. And so, you know, when we try to answer that question, which variety is the best, the answer is often, it depends. It depends on where you grow it. So we have a commercial rootstock here that did pretty well in North Carolina, ranked 4th, but really not very well in Minnesota. Okay, so we can use this kind of fixed effects to try to help us select the best variety. But that same data can be analyzed in a different way, where we're asking a different question. We're no longer asking what's the best. We're asking what are the important sources of variation. This is really the nature-nurture question. Okay, and so we take the same data and the same experimental design, but instead of focusing on the p-value, we focus on the amount of variance explained by the components in the experimental design. And so I'm sharing some results here that came from some work that Sean Fenstermaker did in my group. I'm sorry, I apologize. This first slide is from Nancy Horace's work, where Nancy was looking at components of seed. So fruit set, seed yield, seed weight, and germination. And what I want to point out is that the female parent often plays a very important role. So this is 16% of all the variance is due to female parent for fruit set. Seed weight, that's up to 27%. But environment is also important. So Nancy did her seed production in a winter season and in a spring season. And you can see that environment is just as, if not more important than actual female choice when producing seed. That for seed weight, it looks like genetics outweighs environment, but for these other traits, there's kind of a balance between genetics and environment. Okay, and the next slide shows a similar analysis, and this was Sean's work. So in this case, what Sean was looking at was the variance components for yield, for canopy size, for the formation of adventitious shoots and for sugar content of the fruit. And so what I want to point out, if we look at yield, we'll see that, okay, rootstock has a significant effect. So this is the proportion of total variance. So that would be 0.2%. I'm sorry, it'd be 2%. Whereas the scion is explaining here close to 10%, right? But location is explaining almost a quarter of the total variation. So if we look at these traits, you can see that the relative importance of rootstock and scion is always swayed in the direction of scion being really important. The one exception to that is when we look at these adventitious shoots. And so these are shoots that are coming from the rootstock itself, either from below ground or above ground. And it's a negative trait because it requires either further pruning, and if that pruning isn't done in a timely manner, the rootstock itself will outgrow the scion. So the take home, and I'll come back to that trait in just a little bit. So when we look at variance explained for yield, for adventitious shoots, Brix, and canopy, the take home message is that rootstocks contribute significantly, but relatively little compared to scion, site or location, and other environmental interactions. And really the one exception to that is this adventitious shoots. But I'm going to point out that residual error or uncontrolled environmental interactions is more important than the rootstock itself. Okay. So the other thing that we can do with these data is we can ask questions about the contribution that specific parents are making. So in this analysis, we had to divide it by female parents and male parents, and that's because many of the wild species can't be used to make reciprocal crosses, so we can't evaluate them as a female and a male. And what I want to point out is that for yield, there's maybe one parent that we can say actually has a positive contribution for yield, but most of the wild material, there's really no significant difference between choice, okay? Now, if we look at the next trait, which is this formation of adventitious shoots, you see that there's very little variation in the female parent or the cultivated parent, but when we look at the wild material, what we see is that especially the Habricades, so LA407 and LA2204, these green-fruited wild species, tend to produce more adventitious shoots. So there's a fairly strong genetic signal. This is something we have to be aware about when we choose these parents. Okay. I'm gonna pause and just take a little bit of a segue now. So one of the take-home messages of what I've been saying earlier is that rootstocks contribute significantly, but relatively little to variation compared to scion, site, or other environmental interactions. And so you know, we kind of reinforce that here where there's really no role for different parents and a very small one for the female parent here, okay? So the point then would be that maybe grafting to a rootstock doesn't really help, at least under that soil production open field situation. I feel like it's important that I point out that we have definitely seen exceptions to this comment. And so here we're looking at a scion Cherokee purple, ungrafted, and then grafted to this Ohio rootstock in a high tunnel situation. And you can see that grafted plant is much more vigorous. So we had seen that in the correlations, but in the open field, those correlations of more vigor didn't translate into more yield. Here in the high tunnel situation, we do actually see an increase in yield. But I'll point out that we have season extension here as well, so we can keep these plants around longer and harvest for longer periods of time, okay? So the next exception that I want to mention, when we were evaluating processing type material in the open field, we had a scion, this FG99218, that was selected as a high lycopene scion. So this was actually selected for human nutrition studies looking at how humans absorb lycopene. It turns out that these high lycopene scions have really beautiful deep red fruit due to high lycopene, but they're not very vigorous plants. The lycopene levels are in fact so high that gibberellic acid metabolism is disrupted. The plants tend to be a little stunted. They tend to be sensitive to a lot of foliar diseases in the field, et cetera. So we were interested in knowing whether grafting could improve the performance of this scion without detracting from the quality, which in this case was high lycopene. And so what I want to point out in this slide is that we can, in fact, statistically significantly increase yield across multi-location, multi-year studies relative to ungrafted material, and that in increasing this yield, we don't detract from the high lycopene content that makes that particular scion of some value. Okay, so go on to the next slide. So the summary then is that crop wild relatives or wild species are an excellent source of traits, especially resistance to soil-borne diseases. New rootstocks are available. They've come out of the work that's been featured as a part of this project, and I featured Carolina Strongback as the prime example of that. Genetic signals are low in open field production systems unless the scion is weak, so an heirloom tomato or the high lycopene tomato, unless there is season extension. So there's the value of grafting to a high tunnel or greenhouse situation is higher than to an open field soil-borne situation. And again, the genetic signals for rootstock performance are low unless there is a specific soil-borne disease and a resistant rootstock. So there are certainly places to use rootstocks in an open field production system. And so watermelon production where Phytophthora is present in the soil, Carolina Strongback gives a great management tool under those circumstances. And I'm just going to point out the obvious, but also give a nod towards the groups that have been working on production situation questions, is that site selection and scion choice are still really important. And that's what the variance partitioning really highlights for us. So I want to thank you for your time and again point out that this is work that was done by a number of groups as a part of this project. Thank you very much.